Hi, welcome to Microbiology Session 4. Now, let us discuss about viruses and other acellular infectious agents. During the summer of 2010, over 21 million hot dogs were sold to fans attending games at Major League Baseball parks in the United States. Hot dogs and lunch meats are popular at outings such as baseball games and in lunches carried to work or school. Yet, each year in the United States, approximately 1,600 people are sickened by a bacterium that can contaminate the meat and, even worse, survive and grow when the meat is properly refrigerated. This young man is clearly enjoying his hot dog. Hot dogs and other ready-to-eat meats are a favorite at picnics, baseball games, and other sporting events. However, they can also transmit foodborne pathogens. The disease culprit is Listeria monocytogenes, a gram-positive rod found in soil and many other environmental sites. It is not only cold tolerant but salt and acid tolerant as well, although it is in the minor leagues when compared to some of the big hitters of foodborne diseases like Salmonella enterica. It is of concern for two reasons: who it kills and how many it kills. Listeria monocytogenes targets the young and old, pregnant women, and immunocompromised individuals. About 15% of those infected die. Viruses as agents of goodwill come as a surprise to many. Typically, we think of them as major causes of disease. However, viruses are significant for other reasons. They are vital members of aquatic ecosystems. There, they interact with cellular microbes and contribute to the movement of organic matter from particulate forms to dissolved forms. Bacterial viruses are being used in some European countries to treat infections caused by bacteria, and a number of animal viruses are being used to target and destroy cancer cells. Finally, they are important model organisms. In this lesson, we will introduce viruses and other acellular infectious agents. Viruses are acellular. The discipline of virology studies viruses, a unique group of infectious agents whose distinctiveness resides in their simple, acellular organization and pattern of multiplication. Despite this simplicity, viruses are major causes of disease. For instance, many human diseases are caused by viruses, and more are discovered every year, as demonstrated by the appearance of severe acute respiratory syndrome in 2003 or the SARS, the H1N1 swine influenza virus in 2009, the H7N9 avian influenza virus in 2013, the Middle East Respiratory Syndrome coronavirus or MERS-CoV also in 2013, and the SARS-CoV-2 or Severe Acute Respiratory Syndrome coronavirus 2, which caused or causing the COVID-19, commonly known as coronavirus disease 19. However, the simplicity of virus also made them attractive model organisms. They served as models for understanding DNA replication, RNA synthesis, and protein synthesis. Therefore, the study of viruses has contributed significantly to the discipline of molecular biology. In fact, the field of genetic engineering is based in large part on the use of viruses and viral enzymes, such as the retroviral enzyme reverse transcriptase. Viruses can exist either extracellularly or intracellularly. When extracellular, they are inactive. because they possess few, if any, enzymes and cannot reproduce outside of living cells. When intracellular, viruses exist primarily as nucleic acids that can, at some point in the viral life cycle, commandeer host cells and use them to synthesize viral components, from which progeny virions are assembled and eventually released. Viruses can infect all cell types. Numerous viruses infect bacteria. They are called bacteriophages or phages for short. Fewer archaeal viruses have been identified. Most known viruses infect eukaryotic organisms, including plants, animals, protists, and fungi. Viruses have been classified into numerous families based primarily on genome structure, life cycle, morphology, and genetic relatedness. These families have been designated by the International Committee on Taxonomy of Viruses, the agency responsible for standardizing the classification of viruses. Virion structure is defined by capsid symmetry and presence or absence of an envelope. Virion morphology has been intensely studied over the past decades because of the importance of viruses and the realization that virion structure is simple enough to be understood in detail. Progress has come from the use of several different techniques: electron microscopy, X-ray diffraction, biochemical analysis, and immunology. Although our knowledge is incomplete due to the large number of different viruses, we can discuss the general nature of virion structure. Virions range in size from about 10 to 400 nanometers in diameter. The smallest are a little larger than ribosomes, whereas mimiviruses, the largest viruses known, have virions larger than some of the smallest bacteria and can be seen in the light microscope. However, most virus particles must be viewed with electron microscopes. The general structural properties of virus. The capsid surrounds the viral nucleic acid, protects the viral genome, and often aids in its transfer between host cells. Among the proteins encoded by the viral genome are the capsid proteins, which are called protomers. Capsids self-assemble by a process that is not fully understood. Some viruses use non-capsid proteins as scaffolding upon which the capsids are assembled. Probably the most important advantage of this design strategy is that the viral genome is used with maximum efficiency. Let us take for example the tobacco mosaic virus or TMV capsid is constructed using a single type of protomer. Recall that the building blocks of proteins are amino acids and that each amino acid is encoded by 3 nucleotides, the building blocks of nucleic acids. The TMV protomer is 158 amino acids in length. Therefore, only about 474 nucleotides are required to code for the coat protein. The TMV genome consists of 6,400 nucleotides. Thus, only a small fraction of the genome is used to code for the capsid. Suppose, however, that the TMV capsid were composed of 6 different protomers, all about 150 amino acids in length. If this were the case, about 2,900 of the 6,400 nucleotides in the TMV genome would be required just for capsid construction, and much less genetic material would be available for other purposes. The various morphological types of virions primarily result from the combination of a particular type of capsid symmetry with the presence or absence of an envelope, a lipid layer external to the nucleocapsid. There are 3 types of capsid symmetry: helical, icosahedral, and complex. Viruses with virions having an envelope are called enveloped viruses, whereas those lacking an envelope are called non-enveloped or naked viruses. Tobacco mosaic virus is a well-studied example of helical capsid structure. The self-assembly of TMV protomers into a helical arrangement produces a rigid tube. The capsid encloses an RNA genome, which is wound in a spiral and lies within a groove formed by the protein subunits. Not all helical capsids are as rigid as the TMV capsid. The nucleocapsids of influenza viruses are thin and flexible and are enclosed within an envelope. The size of a helical capsid is influenced by both its protomers and the viral genome. The diameter of the capsid is a function of the size, shape, and interactions of the protomers. The length of the capsid appears to be determined by the nucleic acid because a helical capsid does not extend much beyond the end of the viral genome. The figure shows the schematic view of an influenza virus, which has segmented genomes consisting of 7 to 8 different RNA molecules. Each is coated by capsid proteins to create 7 to 8 flexible nucleocapsid. The envelope has spikes that projects 10 nanometers from the surface at 7 to 8 nanometers intervals. Icosahedral capsids are the most efficient way to enclose a space. They are constructed from ring or knob-shaped assemblages of 5 or 6 protomers. The assemblages are called capsomers. Capsomers composed of 5 protomers are called pentamers or pentons. Hexamers or hexons are capsomers that possess 6 protomers. Pentamers are usually at the vertices of the icosahedron, whereas hexamers generally form its edges and triangular faces. The variants of some RNA viruses have pentamers and hexamers constructed with only one type of subunit. Other variants have pentamers and hexamers composed of different proteins. Although many icosahedral capsids contain both pentamers and hexamers, some have only pentamers. The figure shows the icosahedral capsid of an adenovirus. This shows the electron micrograph of adenovirus variants, which has 252 capsomers. This one is a computer-simulated model of an adenovirus variant. Most viruses have either icosahedral or helical capsids, but some viruses do not fit into either category. Pox viruses and large bacteriophages are two important examples. Pox virus variants are among the largest of the animal viruses, about 400 by 240 by 200 nanometers in size, and can be seen with a light microscope. They possess an exceptionally complex internal structure with an ovoid to brick-shaped exterior. This figure shows the virion morphology of a vaccinia virus. Its double-stranded DNA genome is associated with proteins and contained in the core, which is a central structure shaped like a biconcave disc and surrounded by a membrane. Two lateral bodies lie between the core and the virion's outer envelope. Some large bacteriophages have virions that are even more elaborate than those of pox viruses. The virions of T2, T4, and T6 phages that infect Escherichia coli are said to have vinal symmetry because they have a head that resembles an icosahedron and a tail that is helical. The icosahedral head is elongated by one or two rows of hexamers in the middle, and contains the DNA genome. This figure shows the structure of T4 bacteriophage virion, and this figure shows the electron micrograph of the virion. The tail is composed of a collar joining it to the head, a central hollow tube, a sheath surrounding the tube, and a complex base plate. In T-even phages, the base plate is hexagonal and has a pin and a jointed tail fiber at each corner. The nucleocapsids of many animal viruses, some plant viruses, and at least one bacterial viruses are surrounded by an outer membranous layer called an envelope. Animal virus envelopes usually arise from the plasma or nuclear membranes of the host cell. Envelope lipids and carbohydrates are therefore acquired from the host. In contrast, envelope proteins are coded for by viral genes and may even project from the envelope surface as spikes, which are also called peplomers. In many cases, spikes are involved in virion attachment to the host cell surface. Because spikes differ among viruses, they can also be used to identify some viruses. Many envelope viruses have virions with a somewhat variable shape and are called pleomorphic. However, the envelopes of viruses such as the bullet-shaped rabies viruses are firmly attached to the underlying nucleocapsid and endow the virion with a constant characteristic shape. Influenza virus is a well-studied envelope virus with two types of spikes. Some spikes consist of the enzyme neuraminidase, which functions in the release of mature virions from the host cell. Other spikes are hemagglutinin proteins, so named because they bind virions to red blood cells and cause the cells to clump together, a process called hemagglutination. Influenza virus hemagglutinins participate in virion attachment to host cells. Most of its envelope proteins are glycoproteins, proteins that have carbohydrate attached to them. A non-glycosylated protein, the M protein, is found on the inner surface of the envelope and helps stabilize it. One clear distinction between cellular organisms and viruses is the nature of their genomes. Cellular genomes are always double-stranded DNA. Viruses, on the other hand, employ all four possible nucleic acid types: double-stranded DNA, single-stranded DNA, single-stranded RNA, and double-stranded RNA. All four types are used by animal viruses. Most plant viruses have single-stranded RNA genomes, and most bacterial viruses have double-stranded DNA. The size of viral genomes also varies greatly. Very small genomes are around 4,000 nucleotides, just large enough to code for 3 or 4 proteins. Some viruses save additional space by using overlapping genes. At the other extreme are the genomes of mimiviruses, which infect protists. Relatively few RNA viruses have double-stranded RNA genomes. More common are viruses with single-stranded RNA genomes: polio, tobacco, mosaic, SARS, rabies, mumps, measles, influenza, human immunodeficiency, and bromosaic viruses are all single-stranded RNA viruses. That is all for the general structural properties of the virus. We will have the continuation of the topic in the next session. Stay safe and God bless everyone. Bye!